Genomic Medicine. So thank you again, Steve, for accepting this position, and you are looking forward for high standard impact journals and work to be published in BJ Genomics. I think we have some time if anybody has questions. Yeah, go ahead, please.
I'm a pediatric cardiologist. I hope you can hear me. There's no microphone. I'm very much happy with the genetic studies that you showed, but are we having all the information about to be able to use it properly? Are we going to face this problem? Like going to the mall and looking at the Zara shop in different countries, all over look the same, but behind that is a different story. Yeah. Are we going to use this, or are we going to have all the information that for the next generation of people and scientists to use it?
Yeah, so the question was, how do you use the information?
Do we need all this information for the whole genome, the studies that we're doing? Like I go and I pay $1,500, became my genome study, and I'm facing information that I don't know if I would ever use it or not. Maybe it's depression. What is the science going to have in this discovery? How far are we going there?
Right. So I would argue that I think we're saying the same thing. I think if there is a clinical reason to pursue sequencing, it should be done because it will help in the interpretation of. And I gave you an example on autism, but our center studies everything from cancer to intellectual, everything in between. We have well over 200 different diseases we're working on. Should the healthy population, no one is healthy, but I think the non-disease population have their genome sequenced? Personally speaking, I don't think so, in my opinion. This was a research experiment we've done with the Personal Genome Project because we want to learn about all the different steps along the way, including transferring the information to the population. But a lot of people are doing it. I think an important statistic is when we do a genome sequencing experiment in any different developmental disorder, 50% of the information we find is not relevant to the actual clinical question that came to hand. So from a cost-benefit perspective, we have to do studies of how that impacts the clinics downstream, both in pediatrics and adult. It's a really significant issue. But what I would say is that we've saved many lives doing this, where we've actually have had an autism family, for example, come into a study and we found a colon cancer gene in the child that was inherited from a parent. And within 2 weeks, we can actually save the father's life based on the mutation that was identified. I could give you dozens and dozens of these types of examples. So I think the norm will be that as a tool for scanning or surveillance of the genome in a disease state, this will take hold. But in the general population, I think it's going to play out, specific to that population. So in California, a lot of people are having their genome sequenced. It may not be the case here.
But what about cardiology? How far are we with cardiology?
Cardiology is it depends on the type of diseases that are being studied. I think cardiology spans rare variant diseases to common variant diseases. In our hospital, actually, cardiology and neurology are the ones that are using genomic scanning technologies the most.
Any more questions? Yes, one.
Carlos, yes. Go ahead. Thank
Hi,
you for
Carlos.
the talk, Stephen. Regarding that question, the last question is, I think it's comparable to the MRI. Should we do MRIs to everybody, whole body MRIs? Of course not, because we will find a lot of things that we are not interested to know, and will do more harm than benefit. Anyway, my question is regarding the CMVs. The paper published in Nature in 2006, which was also on the cover, you were one of the authors there, and now in 2015, in a way, it's a similar paper, no? It's a review. Although the definition, the first paper was, I think, the CMVs above 1 megabase. Huh?
Yeah.
Now your definition have changed probably a lot. And also, what similarities and differences are between both papers? Briefly, if you can.
You can read the paper. So the Yeah, the 2006 was, we did Affymetrix 500K array and a tiling path CGH array of the hap maps, 270 hap map samples. That was the first genome map. You're right, the resolution was down to about 500 kilobases at that point. We had a good map at the time, but then in 2010 with the Sanger Center, we published a much higher resolution map where we did tiling path oligonucleotide arrays that got us down to 500 base pairs. So that's been the gold standard to this point, but it was only on 270. That also was on the hap map samples. That was in Nature in 2010. So only 270 samples at the time. This current study, we looked at, I think it was 15 different studies from thousands of different samples. We had Asian populations better covered. There were some from the Middle East, and it's still largely devoid of non-Western populations, but there's better representation. High resolution arrays, and there's also sequence data too. So we have the, it's called 1,000 genomes sequence data in there. So we go down to 50 nucleotides in this current paper. But I'll tell you, it's still a very rudimentary map. We have what we call a stringent map where we got 12% of the genome, copy number variable across these populations, and then a clinically stringent map where it's a little bit lower. I can't remember what it is now, 10% or so. But I think the idea is that we'll continue to catalog this. There's going to be lots of population-specific rare variants that come up that we want to get into the map too. So perhaps 10 years out, we'll do the next generation map. We'll see. So it takes a lot of time to do these comparisons.
Yes, Dr.
Thank you very much for a very interesting, comprehensive presentation. And the thing is that we are actually over here at CGMR working on autism, and we did screen 14 families using array CGH. The thing is that because over here the cost is an issue that we have to consider, and making complete utilization of an exome sequencing still remains an area that needs to be addressed very seriously. So our main focus is right now that since we have found some interesting CNVs, we are rather trying to go into the direction of a PGM towards using more specific targeted sequencing rather than doing the exomes, since we can probably identify some unique mutations in certain category of families.
Yeah.
And that might help us more and rather address the conditions also, explain them in a better way, and utilization of doing this. Because that is a question that keeps coming up, instead of doing exome sequencing. Yeah. So could you shed some light on that?
So it really depends on the disease you're studying. So in our hospital for the cardiomyopathies and for cystic fibrosis, we're using PGM panels, for example, because there are a well-defined set of genes and mutations. So we sequence sets of genes that have been pre-selected. We do lots of samples across those. Autism, the take-home message from our latest paper in Nature Medicine is that's exactly what you should not do, because you have to go genome-wide. You need to look at all the genes. So you can do the microarray experiments under the caveat that you're only going to find certain size variants, of course. And the exome, we have another paper I didn't talk about where we've done microarray and exome on 200 consecutive diagnoses from a physician's office, and we see actually microarray is a better yield than exome sequencing. But whole genome eclipses all of that. So there have been some papers that have gone and just developed technology for resequencing specific candidate genes. They've been published in Science and that. Our paper says that's exactly what you shouldn't do, because you can't predict the mutation because there's so many different genes involved. I understand your situation here. I think you have to do a cost benefit. I would say for autism, you're much better to try to go genome-wide, my opinion. But for other diseases, no, you're right. You should be doing panels.
Okay.
Yes, what about genetics and the whole project? Is there an expectation of data collection in that field or that approach?
Yeah.
Yes. Yeah, epigenetic. Question was epigenetics, and I think this is of course, we were talking about this the other day, and the interpretation of the epigenetics data has been limited, in my opinion, because the tools that have been used are really just targeting the gene regions, and yet one might expect that epigenetics may have the biggest influence on regulatory regions of genes. So that's the importance, I think, again, of doing whole genome sequencing. We're now running methylation arrays on all of the samples we're doing whole genome sequencing on. So again, I think the way to approach this is to do it in a hypothesis-free way and look across the entire genome. Sample size will be critical. I'll come back to the relevance to the journal. I think these studies of 50 families, maybe 100 families, they may not be enough if you're really doing a genome-wide correction type assay. So you need to think about that beforehand. We do have some, I can tell you, we've done now 200 families. We've looked at the methylation profiles, and we found a few, again, rare types of changes occurring in the regulatory regions of some of these genes I mentioned on that slide, the SHANK3 gene, for example. But again, there are rare changes that are playing into this. So it may be that epigenetic type influences are influencing a proportion of autism also. But we have to wait and see.
Thank you.
You mentioned one non-coding RNA. Is the only one founded so far in autism?
There's only one that is kind of an ACMG diagnostic test now. Yeah, this is a great story. I think you probably know. But again, the power of doing genome-wide testing. When we published the 2010 Nature paper describing our consortium analysis of was it 1,600 families or something. My group ran the C&V analysis, and my postdoc, she focused all of the analysis on the gene coding regions because we looked at ref-seq genes. And then when we were writing the paper, I asked her for the list of all the variants. I said, well, what about this, this deletion that keeps coming up? It's actually in 0.75% of our population. There's a 1 million base pair region on XP11.2 where this non-coding RNA skips through this region. It's huge, multiple isoforms. And in fact, that's the most common deletion that we see in males that have autism. So it's affecting the non-coding RNA. We've generated IPS lines on these patients, from these patients. The gene is only expressed in brain. We think it's acting as an epigenetic regulator, actually. We think it's turning on and off through binding DNA expression of other genes. But that is a bona fide valid diagnostic test now. In fact, that's the one gene that we always see associated with autism or autism-like symptoms. So it might be the autism-specific gene.
Also associated with intellectual disability? Or?
Yeah, so we see intellectual disability. We've seen intellectual disability. When we've gone back and looked, they're also on the spectrum.
Dr. Abbas, the last question is.
Thank you. I would like to thank you for this nice presentation. My question, what's the reason behind that in copy number variation, percentage of loss is more than gain? The percentage of loss is more than gain in copy number variation.
Yeah.
The reason behind it. The other question might be said, what is the difference between genomes and genomics?
Okay. So is your question with respect to copy number loss in disease or in the general map, in the copy? In the map, yeah. It's an ascertainment issue. It's a technical issue, artifact actually. It's much easier to call losses because it's a 2 to 1 or 2 to 0 than a gain which becomes 2 to 3. It's a 1/3 ratio. So in fact, we've shown this in many of our papers now. It's actually an artifact of the technology. So there's more gains there still to be discovered. Okay, in the disease setting, we see more pathogenic deletions because the mechanistic haploinsufficiency would be predicted to have more of an impact than going from 2 to 3 copies for most mechanistic diseases. Genomics versus genetics? Genomics and genomes? Well, genomics is the application of technology to study genomes. Genome is your constitutional DNA complement.
One last comment I'd like to make, I'm sure you would agree with me that, looking at your studies, you have to, to do large cohort of families, like for example, 50 to 200, whatever, and more. But I would like to assure people as well for the NPG Genomic Medicine. Okay. I would like to assure people for genomic medicine because this is one of the policies that Steve himself put for the NPG Genomic Medicine. He will accept case reports. So
Thank you very much for reminding me, because it's very important. I think genomic medicine, in many cases, the N is going to be equal to 1, right? You're going to have a very specific variant or set of variants. So I think if you only sequence the genome in a case report, that's not going to be enough. But if you can interpret the data to impact the clinical management, or if it influences the medication they take, kind of like a New England Journal type case report, absolutely. I think there's lots of these. So actually that thyroid hormone receptor mutation I mentioned in the autistic patient, we haven't published that yet. We're testing now thyroxine actually to see if that will have an effect. So if that has a positive effect, that would be a beautiful type paper here. So if you have a small family case report with some type of an application, we'd love to see that.
One of Steve's aims for the NPG Genomic Medicine is to target these studies as well. So bear in mind, don't get scared, basically. You'll be fine with us. Thank you very much, Steve. It's a pleasure to have you here. Thank you very much.
Our next speaker, a very well-known, highly cited professor, Professor Ashok Agarwal. He is the Director of Andrology Center and the Director of Research at the Center for Reproductive Medicine. Professor Agarwal holds these positions at Cleveland Clinic in Cleveland, Ohio, USA, where he is a professor at the Lerner College of Medicine of Case Western Reserve University. Just brief introduction because the time about Ashok. He is now familiar professor with us here for the second year. It's a pleasure to have him with a lot of projects and collaborations, and we are after more projects, inshallah, with him. Professor Agarwal, current research interests are identifying biological markers of oxidative stress, DNA damage, and apoptosis using proteomic research tools and bioinformatics analysis, as well as preserving fertility in patients with cancer. Ashok is actively involved in the laboratory of clinical studies, assessing the efficacy of certain antioxidants in improving the fertility of male patients. Actually, Professor Agarwal has published over 500 publications between peer-reviewed articles, reviews, and obviously over 150 book chapters. Very impressive. And he presented over 750 peer-reviewed articles so far, or maybe more, I'm not sure, at both national and international scientific meetings. So it's a pleasure to welcome Professor Agarwal. So please come. This is the, am I right? So the title is The Global Rise in Male Infertility, Research into the Potential Causes. I'll leave it to you.
Good afternoon. First of all, I want to thank the CGMR and KU for this excellent collaboration with the Nature Publishing Group for their journal. I think this is an excellent news and something that CGMR and all the colleagues there in CGMR must be very proud. I want to congratulate Steve for serving as the first editor-in-chief of this very prestigious journal, and also the colleagues from Nature who have come all the way from London to participate in the ceremonies. So my topic was decided at the last minute. Certainly I'm not anywhere close to where the journal lies. My research is in a different domain. And when I was told that I am also supposed to give a talk, I just put something together. So it's really not something that is being conducted in my lab or in my group, but certainly the things that I will probably bring forward are going to be topics that perhaps can be pursued as areas of research in laboratories and centers here in KU as in any other parts of the world, because those topics are still very enigmatic, very interesting, and remains highly controversial because of the lack of good scientific studies at this time. So infertility is defined as an inability to conceive after at least 1 year of unprotected intercourse, and about 15% of couples, married couples, have difficulty conceiving, and infertility affects about 1 in 6 couples in United States, and about 1/3 of these couples turn up for fertility investigation and discussion together. About 15% of couples in the world are infertile. However, the rate of infertility in different regions of the world are not available. So we conducted a recent study to examine the reporting rates of infertility attributed to male factor around the world. So we looked at different regions of the world and trying to determine excuse me, I'll just see if I can. Okay, so we were very interested to see if we can come up with a way to calculate or project the percentage of male factor infertility in different regions of the world, because the incidence of female factor is quite well reported in many parts of the world, but the male factor remains hidden, and there are very poor amount of information which is available. So we, through various types of projections and calculations, we were able to report that the rates of male factor infertility could range anywhere from 50% in North America and Latin America and in Asia. In Middle East, the rates of infertility could be as much as 60 to 70%. So it is a fairly high rate of increase, rate of male infertility. For purposes of calculation, we looked at, for example, in the United States, if there were 1 million cases of infertility among couples, then about half of those cases would be due to the male factor. And similarly, in the cases of Africa and in Middle East, it's the highest rate of male infertility. The reason is that over the years, the sperm quality is significantly declining, and there is a decline in all the semen parameters, whether it is sperm volume, whether it is sperm concentration. There is a significant decline in all these important parameters. There have been various studies from different regions of the world which have been published, which have shown that there is a significant decline in sperm concentration and sperm morphology, which are the main parameters of semen in human male. And these studies have been reported over the last 20, 30 years, and including very large number of male population. So certainly I think there is a a broader consensus that semen quality in males all over the world, there is a decline, but certainly there is a lack of consensus on the risk factors which are responsible for this decrease in male fertility. Some of the risk factors which have been attributed to play a role in decreasing of semen quality is increasing age, the use of smoking, alcohol, or recreational drug such as marijuana, overweight or underweight, presence of various types of sexually transmitted diseases, diabetes, exposure to various pesticides, presence of varicocele, a condition where there is a vein in the testis region which can heat up the testis by a degree or two and causing decrease in spermatogenesis, prior vasectomy or vasectomy reversal, use of chemotherapy or radiation treatment in patients, or certain medications such as antibiotics and antipsychotics. So I wanted to take up some of these factors and wanted to bring out the available studies to present what kind of evidence there is in the worldwide literature, and you will be amazed that the literature is really very weak and very controversial. And this is an area, as I will go through various risk factors, where well-designed, strong studies can be conducted to provide a sound basis between these risk factors and male factor infertility. So diet is a significant factor, and this study, which was published in Fertility and Sterility, looked at the food intake and its relationship with semen quality. The goal was to compare the dietary habits in normospermic and oligosteroteratospermic men with poor semen quality of both count, motility, and morphology. And it was an observational study, a case control study, and if you look at the number of participants is very small, of just 30 men, and about 31 normospermic controls. And the dietary habits were recorded using a food frequency questionnaire, frequency of intake registered in a scale ranging from no consumption to repeated daily consumption. And what they found was that in oligosteroteratospermic men, oligosteroteratospermic is basically patients with poor semen quality. Oligo means lower sperm count, poor sperm motility, and poor sperm morphology. They had a lower intake of vegetable and fruits and higher intake of dairy and meat products. And they concluded that frequent intake of lipophilic foods, example meat and milk, may negatively affect semen quality, while fruits or vegetables may maintain or improve semen quality. Another study looked at dietary and semen quality analyzed for 99 men at Massachusetts General Hospital Fertility Center. And what they found in this study, that high total unsaturated and monosaturated fat intake is associated with lower sperm count. Again, the high total saturated and monosaturated fat is associated with lower sperm count, and the higher omega-3 intake is associated with improved sperm morphology, according to these authors. Another study looked called Rochester Young Men's Study, about 188 men aged between 18 and 22 years of reproductive age, administered food frequency questionnaire, and two dietary patterns were identified by factor analysis. Western diet, which was a high intake of red processed meat, refined grains, pizza, snacks, high energy drinks, and sweets. Prudent high intake of fish, chicken, fruit, vegetable, and whole grains. And what they found was that the prudent diet was positively associated with the sperm motility in multivariate models. Men in the highest quartile of the prudent diet had 11% higher percentage of progressively motile sperm compared with lowest quartile men. But Western pattern was not associated with any other semen parameters. Now moving on to exercise and fertility, which is another important issue. This study looked at physically active men, whether they show better semen parameters and hormone values than sedentary men. The size of the study, as you can see, we looked at the literature to find the best studies that we can. So these are a representation of some of the studies which we could find, which provided the best type of information. Certainly, there is a great lack of good solid studies. So this was just a 31 healthy male volunteers which were without infertility, divided into two groups. Physically active, had practiced endurance activities except bicycling for over a year, and sedentary, had not had regular physical activity during the previous year. And what these men demonstrated was that active men had greater sperm velocity, higher normal sperm morphology, higher levels of both FSH, LH, and testosterone, and higher testosterone and cortisol ratio, indicative of anabolic state. Their study looked at 189 men, 18 to 22 years, and their physical activity was assessed via questionnaire as to how many hours per week of moderate or vigorous exercise was done, whether they were watching TV for a number of hours a week. And the study found that sedentary lifestyle was associated with the sperm concentration and total sperm count were directly related to physical activity after multivariate adjustment. Men in highest quartile of activity, more than 15 hours per week, had 73% higher sperm concentration than lowest quartile. Hours of TV watching was inversely associated with the sperm concentration and total sperm count. Now going on to obesity and fertility, which is a problem of worldwide proportion. And there are a number of studies again associating obesity with sperm quality and fertility. And this is one of the studies which was published several years ago, which looked at the semen parameter, blood work, and BMI in about a population of 2,000 men. And what they found was that serum testosterone and inhibin B concentration were about 25% to 32% lower in obese men compared to normal weight men, but there was no reduction in sperm count in obese men. Another study published which looked at the obesity and semen quality and endocrine values, and they found that the semen samples and serum levels of FSH, LH, testosterone, and prolactin in over 2,000 men, and patients were stratified by BMI into 4 groups. And the lower serum testosterone, LH, and prolactin were associated with BMI, but there was no statistically significant relationship between sperm quality and BMI. Another study looked at obesity and fertility, and this was the review of the Danish National Birth Cohort, which reveals that among men with a BMI of greater than 18 kilograms, there exists a dose-response relationship between increasing BMI and subsequent fecundity, time to pregnancy greater than 12 months, odds ratio of 1.2. So this is another study published in Fertility Sterility, which is a retrospective analysis of ICSI, which is one of the assisted reproductive technology. Outcome demonstrated that the increased paternal BMI is associated with decreased blastocyst development, decreased clinical pregnancy rate, and decreased live birth outcome. Now moving on to substance abuse and fertility. Substance abuse, we have things like alcohol use, tobacco, marijuana, anabolic steroids, and opiates. So certainly alcohol has been there forever, and dose-dependent impairment of male fertility. It blocks secretion of GnRH and cleavage of GnRH precursor. It also has an inhibitory effect on pituitary, blocking LH production and secretion, direct Leydig cell toxicity, decreased testosterone production, impairs Sertoli cell function, and increases metabolic clearance of testosterone. Less than 4 drinks per day likely have no effect, but heavy consumption may impair male fertility. So this is a study where 100 alcoholics attending fertility clinic, mild less than 40 grams, moderate between 40 to 80, and heavy greater than 80. There was a dose-dependent worsening of semen parameter. Was another study look at 66 alcoholic attending rehab. They were drinking more than 180 ml whiskey per day. 30 controls. Alcoholic had significantly worse quality of semen parameters. There have been a number of studies on tobacco and its impact on fertility. Of course, we know tobacco has a number of very harmful effects on human health. In terms of the fertility, especially on the semen quality, it compromises oxygen delivery of testes, interference with high metabolic requirements of spermatogenesis. There are over 7,000 mutagens and metabolites in cigarette smoke which have been known, and through the process of oxidative stress, it causes various kind of damage to the cell. And this study was a meta-analysis which looked at the socio-psycho-behavioral factors and male semen quality. And the study included pooled data from 57 studies show that smoking is associated with deterioration of semen quality, including sperm density, count, motility, and morphology. Another study where smokers were significantly associated with lower hypoosmotic swelling test. This is a membrane viability test looking at the viability of the integrity of the sperm membrane of the sperm, lower seminal zinc level, higher DNA damage to the sperm, and higher seminal reactive oxygen species production. Marijuana, which is legalized in Colorado and some other places now in the United States, is being used in many other parts of the world as well. The active ingredient is THC, half-life of between 24 to 36 hours. It is endocannabinoids, a receptor in brain, testis, and sperm, has a short half-life. It suppresses the LH and inhibits sperm mitochondrial activity, decreases motility, and increases apoptosis. So again, this is a compound which has been used for recreational purposes. The sperm were incubated with THC at concentration equivalent to therapeutic and recreational plasma level. And as you can see, there has been a decrease in sperm motility with the increase in the concentration of THC. Anabolic steroids, has second highest rate of abuse, and it has a dose-dependent effects, increases the prolactin, and decreases the LH and testosterone, decreases libido. It has a number of effects, harmful effects, as well as have abnormal semen quality and most often reduction in the sperm motility. This is a study which was published some time ago, and the main findings of the study was the azoospermia is very common in these men. Azoospermia means lack of any sperm being produced, and it is usually reversible between 3 to 6 months, but may take years, and occasionally it is irreversible. So let's move on to environmental factors and fertility. Environmental factors could be cell phone radiation, pollution, as well as heat exposure. So there have been widespread use of cell phone. It's like a necessary part of our lifestyle, but it is also known that the cell phones generate a small amount of radio frequency electromagnetic radiation, and there are a number of studies that have been conducted. Most of them are observational studies at this time, which have looked at the impact of cell phone radiation with male fertility. This is again one observational studies that we published. This was the first one in the literature, where we looked at over 300 men undergoing infertility evaluation divided into 4 groups according to reported cell phone use, self-reporting them. And we found inverse relationship with all the important sperm parameters such as sperm count, motility, viability, and morphology. And there was a dose-dependent as much, so the higher the use of cell phone, the poorer the quality of the semen in these men. Another study looked at daily transmission time of using the cell phone in about 300 men, and they correlated negatively with proportion of rapid progressive total motile sperm, positively with proportion of slow progressive motile sperm. Low and high transmitter group differed in the proportion of rapid progressive motile sperm. There was a significant decrease. Another study which was conducted in our program several years ago was an in vitro study on human semen, which suggested that electromagnetic radiation exerts deleterious effects on sperm function. We had 23 normal healthy donors and 9 infertile men divided into 2 aliquots, an experimental aliquot exposed to cell phone radiation for 1 hour and a control group. The radiofrequency electromagnetic radiation exposure caused decrease in sperm motility, decrease in sperm viability, and increase in the levels of reactive oxygen species. Another study looked at the cell phone use, and they found that the levels, actually this is wrong title, it is the effect of air pollution, and what they found was that the levels of air pollution over a 5-year period compared to sperm parameter obtained from about 1600 semen analysis, and the values of particulate matter were negatively correlated to sperm motility and morphology. Another study looked at this is effect of heat and semen parameters in normal zoospermic men. As you can see, the number of men in these studies is relatively small. Human reproduction is a very highly cited, one of the topmost journal in our field of reproductive medicine. And you can imagine that the study had only 10 men, underwent 2 sauna sessions per week for 3 months, 80 to 90 degree centigrade. And they looked at sex hormones, sperm parameters, sperm chromatin structure, sperm apoptosis, and expression of gene involved in heat stress and hypoxia with sauna exposure, and 3 and 6 months later. Exposure to sauna or extreme heat associated with poor sperm count, poor motility, lower percentage of sperm with normal chromatin condensation, decreased percentage of sperm with normal mitochondrial function, no significant change in sex hormone. All effects were completely reversed 6 months later. Various studies have shown temperature changes in the scrotum are associated with prolonged driving, sitting, wearing restrictive clothing or tight-fitting underwears, laptop or computer use, and use of electric blankets. So I just wanted to conclude this short presentation with some thoughts. Essentially, as you saw, various factors which have been associated as risk factors for male fertility, which is a significant global issue. You will see that still we are in 21st century, but the type of studies being conducted and published relatively to the literature at this time is very poor. We have really no solid evidence to say one way or another that these risk factors really play an important role, and there is a great need for well-designed, good studies that should come out in the literature to allow to